Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear students, now we are studying class 11th chapter, that is biological classification. In this lecture series of biological classification, we have already completed the initial classification systems like two kingdom, three kingdom, four kingdom, five kingdom, six kingdom, and three domain classification system. And then we have studied kingdom Monera, in which we have also studied the different specifically structures, different life processes, and the different types of bacteria which included in kingdom Monera. Now, after this, in biological classification, we are going to study a next kingdom, that is kingdom Protista. Dear student, this is a kingdom in this study. This is specifically kingdom Protista, and kingdom Protista is specifically known as kingdom of unicellular eukaryotes. Okay, so they are particularly started with the study of unicellular eukaryotes, which are including kingdom Protista. Now, in this kingdom Protista, we will be talking about the Protista cells, which are eukaryotic, which have a well-defined nucleus. If they are having a eukaryotic structural organization, then obviously they have a well-defined nucleus. They have a membrane-bound organelles. Now, in case of this protistans, it is known that flagella or cilia are present in some members of protists. So we will see that there are some protists in which the flagella or cilia are absent, and we will also study some examples in which we will find that the cilia and flagella are specifically absent. Okay, so these are some of the diagrammatical representation of this protistans, which are unicellular eukaryotes. Now, when we talk about this particularly protist, it is known that they reproduce asexually by fission, means asexual reproduction they perform, and that is by fusion or fission or by the formation of spore. Now, when sexual reproduction occur, that involve a cell fusion and zygote formation, means the cell are going to fuse. So, but the important thing is that the boundaries of this kingdom are not well defined. Boundaries of this kingdom are not well defined. So the question is why? Because this members of kingdom Protista forms a link with the other kingdoms dealing with the plant, animal, and fungi. So if this particular kingdom has some characteristic that is forming a link with the other three kingdoms, means it has some characteristic of plant, animal, and fungi. So the boundaries are not clearly defined because they have fungal character, plant character, as well as animal character at some extent. This is the life cycle in which you will see the zygotic meiosis. In this zygotic meiosis, dear student, you can see that this is the particularly gametes. Okay, and in these gametes, the young individual develop from the gamete that is haploid, then an adult will be haploid. Then adult undergo mitosis and gametes are produced, that are two haploid gametes. Then fertilization will occur, and the zygote is going to form. So this is the thing. Now, so single cell eukaryote are placed under this protista. These protista are primarily aquatic. They are usually aquatic. So they are primarily aquatic, but we will be dealing about this protista and their characteristics in detail. So when we talk about some characteristic of protistans, they have some of the particular unicellular organization. But just we have studied about the reproduction, I want to make you understand something in reproduction of this protist in detail. If they are particularly protists, so we have to deal with the reproduction of protist in some more detail. In reproduction, they perform asexual reproduction, and in this asexual reproduction, there occur a binary fusion. And this binary fusion where daughter cells are formed by the division of one mother cell. Here in this, one mother cell is there that is divided into the two. After these two daughter cells or each daughter cells grow to form a new individual. And these are also in unicellular, so it will grow and act as a new individual. So two individuals will be formed by this binary fusion. And this binary fission is known to occur in case of chrysophytes, dinoflagellates, and immunoids. In this asexual reproduction, spore formation also occur. Spore formation also occur. So, for spore formation, this protists have a special structure known as sporangia, and these spores are formed within sporangia. And when this sporangium bursts, after some time, all the spores are released and they become free. And when each spore finds its suitable substratum that is going to form a new member. So this spore are formed within sporangium. Okay. Another type of reproduction after asexual reproduction is sexual reproduction. And this sexual reproduction was first of all seen in protists. First of all seen in protist. Now, in this sexual reproduction, here two haploid gamete fuse and form a diploid zygote. Here, two haploid gamete, one can be considered as male gamete, another one can be considered as female gamete, and here fusion occurs and diploid zygote is formed. Diploid zygote is formed. And this fusion is known as syngamy. What it is known as? It is known as syngamy. And in sexual reproduction, this syngamy is of several types, and actually it is of three types. So next we are going to study the three types of syngamy. So when we study this syngamy, This syngamy is of three types. In this, the first type is isogamy. And isogamy is the simplest way of sexual reproduction. And in isogamy, the fusing gametes are morphologically similar. This in isogamy, fusing gametes are morphologically similar, but physiologically so when morphologically means in the shape, size and structure if gametes are similar, but in physiology, in function or in genetic level if they are dissimilar, then this type of fusion is known as isogamy. So this isogamy show a dissimilarity in physiology, so it is also called as or also known as physiological anisogamy. Then another type of syngamy is that is anisogamy. And in anisogamy, the fusing gametes are morphologically as well as physiologically dissimilar. If the fusing gametes are morphologically as well as physiologically dissimilar, means both male and female gametes are dissimilar in size, shape, as well as internal genetics is also different. So it is known as an isogamy. And the third type which you can see here, we are going to study, that is oogamy. And in oogamy, it is known that it is a developed form of anisogamy. It is developed form of anisogamy. Here, male gamete are here male gamete are small and motile, whereas the female gamete is large and non-motile. And this female gamete is known as egg. So if such type of gametes are there and their fusion occur, then it is known as oogamy. Here these gametes are formed within sex organ, and here within the sex organ where the female gamete is present, male gamete will reach to that particular part and the fertilization will occur within the sex organ. So it is a developed form of anisogamy. It is the oogamy. So these three types of syngamy can occur during the sexual reproduction in case of a protist. Now, We have studied that these are the single cell eukaryote, they are primarily aquatic. Now, what are the members? Which are the members of this protista? So we have to talk about the protista members, and if we talk about the protista members, this include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. This kingdom also forms a link with other like plant, animal, and fungi, and that we have studied. Now, dear students, we are going to study this all organism one by one. But in this, we remember this chrysophytes is going to include diatoms and desmids. And in this, the three are there: the chrysophytes, dinoflagellates, and euglenoids. And when these three are included, they are included in one group that is known as photosynthetic protist. This photosynthetic protist, if they are included in this all are capable of the photosynthesis. But this slime fold is the another group of organisms which are placed in case of this protist, but they do not have the capacity to perform photosynthesis. So they are known as consumer decomposer protist. They are known as consumer decomposer protist. Because when we talk about nutrition, majority time the nutrition of slime mold is saprobic or we can say phagotropic. So they are a consumer decomposer protist. Then we will be going to talk about the protozoans, which is the last group of organism which show particularly heterotrophic nutrition, and these are placed in this group. So we are going to first study this photosynthetic protist, then we will talk about consumer decomposer protist. Now when we will study all this particular organism, first we talk about the chrysophytes in that, and chrysophytes include diatoms and desmids. Desmids are considered as a golden algae due to their color. Now, when we talk about the chrysophytes and their habitat, it is known that they are found in freshwater as well as in marine environment. The habitat is freshwater as well as marine. So you can see these are the different particular examples of chrysophytes. In this chrysophytes, we can understand that these are specifically of different shape and they are found in such a variety of ways. Okay. In these chrysophytes are so microscopic that they float passively in water current. So they become a part of what? This planktons. They become part of this planktons. And when they are become a part of this planktons, you can see this is the different shape of these diatoms. Now, most of the chrysophytes are photosynthetic. They have pigments, so they are able to perform photosynthesis. So because of this, they are placed in particularly this group that is photosynthetic protist. Now, in diatoms, the cell wall form two thin overlapping shells known as epitheca and hypotheca. In diatoms, actually, when we see the body has one upper cell and the lower cell. Upper shell is known as epitheca and lower shell is known as hypotheca. You can say this is the diagram. And by combination of this, they give a soapbox arrangement. They both use a soapbox arrangement. Okay. So this gives soapbox arrangement. You can see this is the upper shell hypotheca, lower shell hypotheca, upper shell epitheca. And when we see the shells of this particular diatoms in their walls, we found that the wall is embedded with silica, and due to the presence of silica, the cell walls are indestructible. One of the important features that you should remember: the cell wall of these atoms are indestructible because they are made up of silica. You can see this particular diagrammatically as well. Now, when we talk about other characteristic, we came to know that they are found in different shapes like circular, rectangular, triangular, elongated, and boat shape. Now, in their cell wall, if we see, it is not like that only silica is present, but in their cell wall, cellulose is present and silica particles are embedded within. Okay, and due to presence of silica, their cell wall is known as siliceous cell wall or silicated cell wall. And this siliceous cell wall are known as frustule. They are known as frustule or shell. In this, you know that just we have studied, they have a upper shell and the lower shell. Upper shell is known as epitheca and lower shell is known as hypotheca. These both are secreted by cell itself. Okay, so in such a way, the cell wall is made up of two halves, which is giving a soap box like structure. That cell walls are not get decomposed after their death, so at the bottom of sea you will find their accumulation. Now, when we keep them in the photosynthetic group, they have some pigments. And when we talk about the pigments of these diatoms, they have chlorophyll A, chlorophyll C, and they have a xanthophyll as well. And in xanthophyll, they have fucoxanthin. And due to the pigments, they are they are able to photosynthesize. And it is known that they something produce about 50% of the production of food is done by this one. Because of this, they are known as chief producer of ocean. And in Neet, I think two times the same question asked in Neet, that which organism are known as chief producer of ocean, then we remember they are nothing but which one? Chrysophytes. And after performing the photosynthesis, they have ability to food store, and the storage food, if we talk, which is found in this, that is leucocene, and this leucocene is known as chrysolaminarin. Apart from chrysolaminarin, which is a carbohydrate, oil are also found as stored food in some of the diatoms. After the storage of food, Next, when we talk about the movement in case of diatom, it is found that they are usually immobile. Why they are immobile? Because flagella is absent throughout the lifespan. The flagella is absent throughout the lifespan, so they are not going to move through the flagella, so they are immobile. But they can float passively on surface of water. Means if water is flowing and with water if they flow also, then this flow movement of this chrysophytes is known as passive movement. Apart from this, it is known that they also show movement by mucilage propulsion. In mucilage propulsion, it is known that suppose this is a one diatom and this is a front portion and this is a back side, and from this, if this is particularly releasing a mucilage by force, so release mucilage at back side by force, and due to release of this mucilage released by force, according to the Newton's third law, they are going to also feel a one force, and due to which they will move forward. So due to the release of these mucilage, they get a force and they move somewhat. So because of this, they are known as what? It is known as mucilage propulsion. Now, after this movement, next we are going to talk about the reproduction in case of these diatoms or chrysophytes. Now, when we talk about the reproduction, it is known that they perform both asexual and sexual reproduction. So in this, reproduction is of two types: asexual and sexual. In asexual type of reproduction, they can perform a binary fission, and this binary fission is occurring in favorable condition, and in that specific also they are show a transverse binary fission. Apart from this binary fission, they also show a spore formation in unfavorable condition. But when we talk about sexual reproduction, in this they show isogamy, anisogamy, and oogamy. In different example, different type of this thingamy occurs, but that ultimately lead to the formation of zygote. And this zygote, which is formed after this, that is diploid, and that zygote is known as oxospore. So oxospore is a special type of spore which is formed during a sexual reproduction in case of this chrysophytes. Okay, so these are the particular certain features which are associated with chrysolites. Now, when we talk about the further detail, it is known that in diatoms, the diplontic life cycle is found. Here, if we talk about the life cycle in case of diatoms, the diplontic life cycle is found. And this diplontic life cycle is also known as gametic meiosis. It is known as gametic meiosis. And this gametic meiosis means the meiosis or reductional division occur to produce a gamete. Otherwise, majority stages in life cycle are diploid. Suppose if we want to draw the diplontic life cycle, we can draw like this. Suppose the diploid zygote is formed, and this zygote is a single cell. It undergo growth, and when it undergo growth, it is going to form again a diploid grown cell, which will be known as adult. Now, this adult single cell is going to undergo meiosis, and in this meiosis, two haploid cells are produced. These two haploid cells again fertilize or again undergo syngamy, and this syngamy or fertilization is going to form a zygote. Okay, so this is the type of life cycle, a diplontic life cycle, which is also known as gametic meiosis, found within this diatoms. Now, after life cycle, we are going to talk about some of the uses of this diatoms. And when we analyze some of the uses of these diatoms, we come to know that these diatoms are also used in soundproofing because they are used to prepare soundproof glasses. So soundproof houses can also be produced. Apart from this, they are also used in filtration of oil and syrup. Then they are also used in stone polishing. They are also used in cup syrups also. We have studied this. They are also used in nail polish remover. They are also used in paints to increase their night visibility. So these are the some of the particular uses, and it can also act as a heat insulator, in stem boilers. steam boilers actually. So these are some of the uses of this particular items. So I hope you now understood this properly in detail. Now I am reviewing this so we can understand it properly. So now the next thing in diatoms which we are going to study, that is the diatomaceous earth or castle. Dear students, when we talk about a diatomaceous earth or castle, we have studied that walls of these diatoms are indestructible. So if walls of these diatoms are indestructible, they get accumulate at the base of this particular ocean or sea. So the accumulation of large amount of silica in the cell walls of diatoms referred as diatomaceous earth, and this is obtained by the mining of this particularly but accumulated indestructible cells of silica because they are not going to decompose. You can see this is one of the diagram of microscopic view. Then this accumulation over a billion years was referred as diatomaceous earth. So suppose if you see this is a water body and below this water body the accumulation occurred and we call this accumulated land as a diatomaceous earth or diatomite or calcareous. Now this can be obtained through mining and it is known that through mining whitish powder is obtained and this whitish powder is also known as Kaisalgar diatomaceous earth, that is this one, Kaisalgar or diatomaceous earth or diatomite. Now, this mined diatomaceous earth is used in several things that we have discussed. Apart from this, diatoms are known to produce a food, so they are known as chief producer of ocean, which we have studied already. The soil of diatomaceous earth being used in polishing, filtration of oil syrup, that also we have discussed. Okay, so that is all about the particularly specifically about the diatoms, chrysophytes, or we can say they are these diatoms and desmids are placed in chrysophyta. So after this chrysophytes, we are going to talk about another photosynthetic protist, that is dinoflagellates. In dinoflagellates, it is known that mostly they are marine and they are photosynthetic protists, so they can perform a photosynthesis. So this is the diagrammatic representation of this dinoflagellates. You can see like this dinoflagellates are seen when we see in a microscopic view. The depending on the main pigment present in the cells of dinoflagellates appear in different colors. Yes, it is known that these dinoflagellates, which are found on the surface of water, they appear in different color like yellow, green, brown, blue, or red, depending on the main pigment in their cell wall. Then you can see the different color can be green, brown, blue, red, yellow, etc. Then when we see the cells of dinoflagellates, the overall on the surface they have a cell wall. And this cell wall are made up of stiff cellulosic plates on the outer surface. So one of the important point that you need to mark that here only cellulose is not present in the particularly cell wall, but cellulose is present in the form of cellulosic plates. Now, usually the name given is dinoflagellates. Dino means two, means they have two flagella, and out of these two flagella, one present longitudinally and the other present transversely. So you can see this is the beautifully represented in the diagram, but here you are not seeing the two particular flagella. But yes, here you can see. Here you can see this is the particularly dissimilar apical flagella. One is this one, the one is the another one. Okay, so this way two flagella are present. These two flagella are present at right angle to each other. So what happens? Both beat at the same time, and due to this, the particularly flagella show one spinning like movement. So this is known as spinning movement. And due to the spinning movement, this protists are also called as the whirling whips. What they are known as whirling whips. So in this question they may ask that which protists are known as whirling whips. Then you should remember they are the dinoflagellates. Then when we see the dinoflagellates internal structure within a cell, they have a condensed chromosome in the nucleus. Dear student, you should remember this, which is this is the one of the important point. They have condensed chromosome within the nucleus. And one of the important thing is that chromosomes do not have histones. Means histones are absent. And this is called as mesokaryonic condition. This is known as mesokaryonic condition. Okay. So now we are going to study some of the characteristics of this dinoflagellates in some more detail. The nutrition you know that is photosynthetic, and photosynthetic nutrition is also known as holophytic nutrition. Okay, so holophytic means photosynthetic. Now when we see some more characteristics which we have discussed now, due to the cellulosic plates, they are also known as, these dinoflagellates are also known as armored dinoflagellates. Now why they are known as armored dinoflagellates? Armored dinoflagellates means the presence of army-like structure, net-like structure due to the presence of these cellulosic plates in their cell wall. Just we have studied that they have two flagella. You can see, suppose this is a hypothetical diagram in which you can see one on the transverse groove and one longitudinal groove. Now, when we see this transverse and longitudinal groove, we come to know that this transverse groove have one flagella and the longitudinal groove have also one flagella. Here, when there is a presence of this two groove and the two flagella in these grooves, dear students, actually this is a transverse groove. See, groove means what? A depression. So this is the transverse groove, and this transverse groove is also known as cingulum annulus or girdle. What it is known as? cingulum, annulus, or girdle. Whereas this is the longitudinal groove, and this longitudinal groove is known as sulcus. And these two grooves have one two flagella. The transverse flagellum lies in the circular transverse groove, whereas the longitudinal flagellum is narrow and it is directed posteriorly. See, it is directed posteriorly. Now, the wall which they have, this thick wall is known as theca or lorica. And in this theca or lorica, there is a pore. Through this pores, this flagella come outside. Okay. Then, if we talk about some more characteristic, like suppose they are photosynthetic, then which pigments are present. Then, if we talk about the pigments, it is known that they have chlorophyll A, chlorophyll C, and xanthophyll. They have a xanthophyll. Then, in these, when we talk about after photosynthesis, the food is stored, and that stored food is known as reserve food. This reserve food is in the form of carbohydrate and oil. Now, in this, we have studied that they have a mesokaryon condition in which the nucleus is large and have a condensed chromosome even at interphase. It does not have histone. Such arrangement is known as mesokaryon. Then they have one more thing that is known as puscle. And puscle is nothing but non-contractile vacuole, which is found in case of this dinoflagellates. And this non-contractile vacuole puscle, it is present near the flagellar base. It is present near base of flagellum. And this particular puzzle may have one or more vesicle. It has one or more vesicle. And if we talk about the function, it function for the flotation and osmoregulation. So it take part in rotation and osmoregulation. Okay. After this, when we talk about the life cycle or reproduction, in reproduction, it is known that they reproduce asexually through cell division. And when we talk about sexual one, In sexual reproduction, they perform isogamy or anisogamy. Then, when we talk about life cycle, in case of dinoflagellates, in case of life cycle, the life cycle is specifically haplontic. Life cycle is haplontic, means zygotic meiosis occurring there. And when we talk about the zygotic meiosis, that is known as the haplontic life cycle. And zygotic meiosis, that is known as haplontic life cycle, which occur in case of some of the examples of dinoflagellates like Serratium and Gymnodinium. Whereas some also have gametic meiosis. Some of the examples also show a life cycle of gametic meiosis. And these examples which show gametic meiosis is Noctiluca. Okay, so this is the life cycle of dinoflagellates. After this life cycle, we have to talk about some of the important events which occur in case of this dinoflagellates. In this, they are responsible for some mechanisms. In this, it is known that they show bioluminescence. The bioluminescence means they emit light, and it is known as, if they are performing bioluminescence and they are emitting light, this, due to the emission of light, they glow at night. And it is shown by some of the examples of dinoflagellates which include Noctiluca, okay, then Pyrodinium. Apart from this, due to the bioluminescence, now how the bioluminescence they cause? Because the cytoplasm have photogenic granule which is responsible for this bioluminescence or emission of light during the night. Because of this bioluminescence, they are also known as fire algae, okay, or night light. Now, apart from this, it is known that they are also responsible for causing a red tide, because red tide. Dear student, if you remember, we have studied Red Sea as well, but it is in case of the cyanobacteria. Now, in red tide, what happens? Some dinoflagellates like Goniolex and Gymnodinium, specifically Goniolex, that proliferate or divide in large number, and due to their proliferation in large number, due to their rapid multiplication in the seawater, this Goniolex spread on the surface of seawater. So, spread on surface of water. Okay, and that particular, due to the spreading of this on the surface of water, seawater appear red, and because of this it is known as red tide. Apart from this, we are going to talk about one more thing which is caused by this dinoflagellates, that is PSP poisoning. Now, dear student, in case of dinoflagellates, they are also responsible for PSP poisoning. Now, PSP means paralytic shellfish poisoning. So the question is, what do you mean by this paralytic shellfish poisoning? Actually, there is one for this causing. There is a one dinoflagellates whose name is Boniolex catenella. It secrete one toxic substance, secrete toxic substance known as saxitoxin. And they secrete a toxic substance that is known as saxitoxin. This saxitoxin, if it is eaten by mussel, that is shellfish, it is one mollusk. So in mussel, it does not cause any particular symptoms. But if this saxitoxin containing mussel are consumed by human, then in human it causes paralysis. Because of this, it is known as paralytic shellfish poisoning. Okay? It is known that these algae also affect marine animals, okay, by causing a water blow. So these are the things with respect to the dinoflagellates. We have studied about the dinoflagellate structure, their habitat, their reserve food, their photosynthetic pigments, and different functions which are shown by them. We have also studied their reproduction. Okay. Now after this, we are going to again note down some of this point that Noctilutica show bioluminescence. You can see this is the emission of light during the night, so it is known as night algae or fire algae. Then red dinoflagellates undergo rapid multiplication and their sea appear red, that is known as red tide. And red tide appear in Mediterranean Sea due to the multiplication of coniolex. You can see this is the red tide which is shown here in this particular diagram. Then the toxin we have just studied, paralytic shellfish. The toxin is produced released by the large number of dinoflagellates that kill marine animals and fishes, not the shellfish. Okay, so this is all about the particularly dinoflagellates. After dinoflagellates, next we are going to talk about euglenoids, a third group of organism in case of photosynthetic protists. Now these are euglenoids are photosynthetic protists and majority of them are freshwater organisms. They are love to grow in a freshwater. And in fresh water also, they prefer to grow in stagnant water. And this question was asked in Neet. Okay, so they are found in stagnant, means stopped water. A particular water is stopped at somewhere, then it is known as stagnant water. Then when we see the cell wall of euglenoid have a protein-rich layer called pellicle. Means we can say like that, the cell wall is absent, the outermost covering is a protein-rich pellicle. And this question was also asked in Neet. proteinaceous pellicle is found in them as outerwear. Due to the absence of cell wall and presence of proteinaceous pellicle, the body of an eukaryote becomes more flexible. And when their body becomes more flexible, the diagrammatic representation of eukaryote is like this. Okay, so we will study this structure as well in detail. Now, just we have a structure, so we note down some of the things in structure in this diagram itself. Dear students, you know that this dinoflagellates have this blunt end. This is the blunt end, and this is a tapering end. Now, in that also, at a blunt end, they have this particular depression, and in this particular depression, if you see this region, if we talk about this region, this is known as mouth. This part is known as cytopharynx. This part, and this is actually a part known as reservoir. In a reservoir, at the base there is a presence of two basal body, and this basal body forms a two flagella. One is long and one is short. But these both two flagella are joined at one point. This is the point where you will find a joining of these two long and short flagella. The long flagella is coming out just like this. Then, at the same level of this particular joining structure, now this joining structure is known as paraflagellar body. So, paraflagellar body is a structure which is a point where long and short flagella meet. And at the same level, on the body of reservoir, you will find a stigma or eye spot. Now here you have to mark, dear student, that this eye spot and paraflagellar body this paraflagellar body both are light sensitive or photosensitive. And in that specifically, if we talk about the eye spot have astaxanthin pigment, which gives some of the reddish coloration. Then in structure we can see one more thing, that they have a nucleolus and nucleus, they have chloroplast, they have pellicle, and they have outermost pellicle, so cell wall is absent. But in place of that, this pellicle is present, and this pellicle is made up of protein. So proteinaceous pellicle is there. They have contractile vacuole as well. Okay, so like this, the structure is there. Now, when we see this structure, that is one of the important structure, we can see, locate some of the important things in case of euglenoids. But when we go on study, we come to know that they have two flagella, one short, one long. We have studied just now anterior part of cell via invagination, consisting of cytostome, that is cell mouth. We have just studied in the particular diagrammatic view in the previous slide that they have a cytostome which is known as mouth, cytopharynx, that is known as gullet, and the reservoir. Photosynthetic stigma or iceberg is present on the membrane of reservoir. That also we have seen in the diagram. Then pigments of euglenoids are identical to those present in plants. The pigments of euglenoids are similar to the higher plants because If we see the pigments which are present in case of glenoids are chlorophyll A, B, and xanthophyll. And this xanthophyll zeaxanthin special type is there, and this pigment are identical to the higher plant. Okay, so after this photosynthesis they store a food, and this stored food is in form of fat and paramylum bodies. This paramylum bodies has a reserve food of beta 1,3 glucan, and this beta 1,3 glucan form a structure that is known as paramylon, and this paramylon is present in paramylum bodies. Now apart from this, next we talk about the movement. When we talk about the movement, these euglenoids are motile. Why they are motile? Because they have, particularly we can say, flagella. So they can be called as flagellated. But apart from this, they also move by metabolic. And this metabolic is a creeping movement due to contraction and relaxation of myomes. Now I can think that you might be thinking like that, what are myomes? So when we study the structure of the euglenoids like this, and they have a long flagella and one short, on their body you will see some of the linings like this, and these linings are known as myomemes, which are present in a pellicle. They can contract and relax, and due to the contraction and relaxation, the creeping movement is observed in case of this euglenoids, which is known as metabolic. Okay, dear students, so these are the some of the characteristics which we have discussed with respect to this euglenoids. Now, if you talk some more thing with respect to this, we have studied that they have contractile vacuole because of this. Now, remember, you can remember one more thing. If contractile vacuole is there, then mostly the organism is freshwater. And usually, whatever the organism which have a non-contractile vacuole, they are mostly marine. You can see, like the dinoflagellates we have studied, they have a non-contractile vacuole, percept. So they are mostly marine. But in euglenoids, they have a contractile vacuole. So they are mostly freshwater. And if they have a contractile vacuole, which occur in the anterior end of the cell, just below the reservoir, and it is meant for the osmoregulation and excretion. They have osmoregulation and excretion. Apart from this, if you talk about reproduction, then in case of reproduction, it is known that they reproduce asexually by mating, that is longitudinal binary fission. And this longitudinal binary fission mainly occur in the favorable condition. Now, apart from this, during unfavorable condition, they form a palmilla stage, they form a cyst, and palmilla stage in unfavorable condition. Apart from this, it is known that sexual reproduction is not known to occur in embryonites. Means these both are the type of specifically asexual reproduction. But sexual reproduction is not known in case of embryonites. This palmilla and cysts which are formed during the unfavorable condition, they are usually made for the perennation to particular carry on or to survive in this what unfavorable condition. Apart from this, it is known that Euglena are known to be producer decomposer protists. Euglena are also known as producer decomposer protist. They are known as producer decomposer protist. They have both characteristic of plant and animal. Why? Because they can perform photosynthesis, so that is a plant character, and they do not have a cell wall, so that is animal character. So they are also known as plant animal. Okay. Then they are known as plant animal because they have a plant-like character and animal-like character both. So if you study plant character which the Euglena have, that are number one, presence of chloroplast, and due to presence of chloroplast they can perform photosynthetic pigment. Presence of chloroplast with photosynthetic pigment. If they have a photosynthetic pigment, then and then only these chloroplasts are of useful kind. They are useful for the what? Photosynthesis. So they carry out photosynthesis, that is holophytic nutrition. Then we talk about some of the animal character or animal-like character which are found in case of this Euglena, and that is absence of cell wall, presence of proteinaceous follicle. Second, presence of stigma, and paraflagelllar body. These both give photosensitive nature, which is an animal-like character. Then presence of contractile vacuole, and then presence of longitudinal body diffusion. This all are animal-like character found in case of Euglena. And both plant character and animal character are present in Euglena, so it is known as plant animal. Okay, so this is the thing. Now, one thing which remained till now, that is nutrition, which question usually you get in this part. And if you talk about nutrition in case of Ulina, they show mixotropic nutrition. Mixotropic nutrition means when light is present or light is available, they absorb it and carry out photosynthesis. But when the light is not present, they perform saprophytic nutrition. So, due to the presence of this dual nature of nutrition, it is known as mixotropic nutrition in case of euglenites. That is all about what are euglenites. In that, dear students, the starting some two characters which was asked in Neet earlier, that is what they are found in stagnant water. They are mostly particularly fresh water. Cell wall is abstent and outermost covering is pedicle. That is proteinaceous in nature. So you should remember this point and all material of euglenites. And here the protista, photosynthetic protists get over. So euglenoids are photosynthetic in the presence of sunlight and heterotrophic in the absence of sunlight. So the reproduction is by longitudinal viable fission and palmella stage. That also we have discussed. Palmella stage is nothing but a immobile round cell which can aggregate in thick jelly layer. Then this immobile round cells aggregate in thick jelly layer. They are known as palmella stage, which is found in euglena. You can see this longitudinal binary division, this division of this particular algae occurring in a longitudinal way, and it will divide into the two body, one this side, one this side. Then you can see this is the palmella stage. In this palmella stage, you can see there is a several particularly cells are found in this. Okay, so dear student, till now we have discussed the photosynthetic protist. Now we will continue giving this protista to the slime mold and then we will discuss the photojournals.